Hello, my name is Lindsay Popp, and I decided to do my project on 3 sets of genes from M. ruber and 3 sets of genes from E. coli, and I found out that each set of genes were orthologs to one another. The first set of genes are M. ruber 2294 and E. coli B2476, which is also known as phosphoribosol aminoimidazole succinylcarboxamide synthase, and the protein known as pure C. The next set is M. ruber 2293 and E. coli B1131, which is also known as adenylosuccinylase, and the protein pure B. The next set is M. ruber 1942 and E. coli B4006, which is also known as ACAR transformylase/IMP cyclohydrolase, and known as the protein pure H. Meiothermus ruber is a unique bacterium in which it grows in high temperature environments that usually range from 35 to 70 degrees Celsius. And not only is it thermophilic, but it is also red pigmented, gram-negative, and non-motile as well. M. ruber was first isolated in the Russian hot springs, and unlike E. coli, there is little known information about M. ruber. While E. coli has over 30,000 sources available, M. ruber has only 28 publications. And while there is such little information known about this unique bacterium, this leads scientists to be able to identify new processes and new genes as we go about learning about this unique bacterium, and it further helps the scientific study and further science in a positive way. The first figure I'm showing is the de novo purine nucleotide biosynthetic pathway that is a part of purine metabolism. As you can see in the figure, the proteins I discussed earlier, pure C, pure B, and pure H, are all involved in this pathway. And they're involved in converting air to fake air, which utilizes the inosine 5 monophosphate IMP compound. And as you can see, the compounds above the arrows are the enzymes that help to convert in this pathway. Overall, this pathway converts the air to the final product of fake air and aids in purine metabolism. The next figure is the purine biosynthesis pathway. This shows the reactants and products that are involved in the pathway, as well as what enzymes are helping to convert each compound to the next. Purine biosynthesis starts with the synthesis of inosine 5 monophosphate, which is known as IMP, because of its versatility to be converted into all other purines. Figure 2 illustrates this pathway where the synthesis occurs. The first step is carried out by pure C, which then moves on to pure B, pure K, pure E, and the final step involves the protein pure H. In E. coli, the first step involves pure K, which is converting air to the then molecule Care by pure E. The next reaction is carried out by pure C, which catalyzes the amino group aspartate to the carboxyl group of Care, and the resulting product forms SACARE. The elimination of the remaining component of the aspartate molecule converts a substrate to fumarate in the next step, which coincides with the formation of ACARE by the protein pure B. The final step involves pure H, which catalyzes ACARE to FACARE, and subsequently to IMP with the elimination of one molecule. As you can see in the first table, this is comparing the bioinformatics tools that were used to show the comparison between E. coli B2476 to the M. ruber 2294 gene. You can see the initial blast resulted in a E-value that was fairly small, showing that there is a high significance that this sequence similarity was not done by random chance alone. As you can see with the CDD category and cog number and name are both identical for the genes. They are both located in the cytoplasm of the cell. The TIGR-FAM protein family and PFAM protein family, name and numbers are identical. However, the PDB showed a difference between the two genes. There are a difference in name and numbers. However, the enzyme commission numbers were identical, and the KEGG pathway number, and name of purine metabolism were identical as well. Also, as you can see throughout the entire table, the e-values are pretty small, which shows, again, the significance of the two genes to one another and a high significance that these two genes are orthologs. And the table is one of the proven pieces of evidence that show that E. coli B2476 and M. ruber 2294 are ortholog genes to one another. The next figure shows the initial protein blast alignment of E. coli protein known as pure C versus the M. ruber 2294 gene. From the results, you can see that 44% of the amino acids were identical, while 141 were similar. The e-value is about 1e to the negative 66, which shows, again, a small e-value, and again shows the significance that the amino acids are not aligned due to random chance alone. The next table is comparing the second set of genes, known as E. coli B1131 and M. ruber 2293. And as you can see, the initial blast gave us a small e-value, which is one of the first pieces of evidence that they are orthologous to one another. The CDD showed an identical number as well as name. They are both located in the cytoplasm. The TIGR-FAM and PFAM protein family information was both identical in name and number. However, the protein database again showed a difference in name and number, but the enzyme commission number as well as the KEGG pathway map and number were identical in that they were located in the purine metabolism map and had the same EC number. As you can see again, the e-values throughout the table are fairly low and show significance that these two genes of E. coli B1131 and M. ruber 2293 are orthologs to one another. The next figure is showing where the E. coli B1131 and M. ruber 2293 genes are located in the KEGG pathway map. Here is the map of purine metabolism, and as you can see, the above panel is E. coli, and the below panel, lettered B, is M. ruber, and they are both located in the same location and conversion step, as well as have the same EC number. In the final table, comparing the last two sets of genes, you can see that E. coli B4006, also known as the protein pure H, and M. ruber 1942 are orthologs to one another. The initial blast search gave a really small e-value. The CDD data was identical in number and name. They are both located in the cytoplasm. The TIGR-FAM and PFAM proteins were both identical in name and number, and the protein database was identical in name and number as well. The enzyme conversion number, as well as the name and KEGG pathway map and name, were identical to one another, and again, the e-values were very small throughout the entire table, showing that the two are orthologs of one another and are similar in sequence, and there is a high significance that the sequence alignment and similarity was not shown due to random chance alone. In conclusion, my results showed that the E. coli gene B2476 and M. ruber gene 2294 are orthologous genes. Again, E. coli B1131 and M. ruber 2293 are orthologous genes. E. coli B4006 and M. ruber 1942 are orthologous genes as well. In the initial blast search, it showed sequence similarity between the two, as well as cellular location bioinformatics tools further confirmed the results that they were identical to one another. The ones we used were TMH, Signal P, Lipo-P, Phobius, and P-sort B. Analysis of the TMH led to conclusions that the proteins did not have a transmembrane helices and showed that the first set of proteins did not have TMH as well and did not have any cleavage sites when looking at the Signal P data. TIGR-FAM and PFAM were able to identify similar protein sequences among the first set of genes, and the phylogenetic tree for M. ruber was significant, showing that there was a common ancestry because both were part of either the Firmicutes or the Meiothermus phylum. By comparing the two first set of genes of B2476 and M. Ruber 2294, it showed similar sequences in comparison to the web logo and PFAM modules. Both genes were located inside the cytoplasm and had identical cog numbers as well as Tiger fam numbers and names. Both were located in the same KEGG pathway map, which was purine metabolism, and also both shared common ancestry. In the second set of genes of E. coli B1131 and M. Ruber 2293, it showed similar sequences in comparison when looking at the web logo and PFAM modules. Both genes were located inside the cytoplasm and had no transmembrane helices nor cleavage sites. Both were identical in cog numbers and Tiger fam numbers and names, and located in the same KEGG pathway map and location of the map, which was again purine metabolism. Also, both shared common ancestry in the phylogenetic trees by containing a Firmicutes and Meiothermus phylum. The last set of genes, E. coli B4006 and M. Ruber 1942, shared similar sequences in comparison to the web logo and PFAM modules. Both genes were located inside the cytoplasm and had no transmembrane helices nor cleavage sites. Both set of genes had identical cog numbers and Tiger fam numbers and names, and both were located in the same KEGG pathway map, which again was known as purine metabolism. Also, both shared common ancestry in the phylogenetic trees by containing a Firmicutes and Meiothermus phylum. Based upon the abundance of similarity found in all of the bioinformatics tools, we can conclude that these 6 set of genes in comparison to one another are orthologous genes. In my SDM, I changed leucine to proline because of the important gene function it had in M. Ruber 2294, and it showed how conserved the protein was and how the function was changed once the mutation was made. In conclusion, I can conclude that E. coli B2476 and M. Ruber 2294 are orthologous genes. E. coli B1131 and M. Ruber 2293 are orthologous genes, and E. coli B4006 and M. Ruber 1942 are orthologous genes to one another. Thank you.